So we will talk about start vectors in molecular biology. Vectors in molecular biology. So what is a vector? Vector actually it is a DNA molecule. It is a DNA molecule. It can be a plasmid or it can be a virus, it can be a plasmid, it is used as a vehicle. Vehicle is a car to transport from one side to another place. This is used as a vehicle. To carry what? Vehicle to carry a DNA segment. Some people may call it as a DNA of interest carried into a host cell. Host cell. This host cell is mostly it is bacteria. Specifically E. coli. E. coli is used most widely. So, a DNA of interest is being carried from outside into a cell, and this process is known as transformation. It is a part of a molecular biology or recombinant DNA strategy, recombinant DNA technology. So, essentially, you have a DNA, circular DNA mostly. It's a DNA molecule in which you have put your DNA of interest, whatever it can be. It can be from that organism, it can be from another organism. You have put it inside this particular circular DNA, and this circular DNA acts like a car or vehicle. It takes that DNA into another cell, host cell. So, if you just look at it, say this is the DNA of interest. And this site here and this site here, they are restriction enzyme sites. So you have got a restriction enzyme site. So restriction enzymes are enzymes which cut at specific sequence on a DNA. For example, EcoR1 is a restriction enzyme. It will always cut GAA-TTC. GAA-TTC. This is a double-stranded DNA. It will always cut at this point. Result, you will always have an overhang like this, AATTC, one side, and here you will have G, and on the other side you will have CCC, AA. This is the other side, and you will have G. CTCAA, sorry. This direction, whatever you see, this direction is also the same. So this is like madam. Both sides you read, it's the same. It's this type of sequence is known as palindrome. Palindrome, exactly. So the ends are like this. So you have an overhang. This is known as overhang. It was soon as a double stranded, then you cut it. You have this region AATT. This region is hanging. एक साइड में G है, कट गया यहाँ पे. So G and you have the G opposite is C. Then you have TTAA. This is known as overhang. This region is known as overhang. ठीक है? To this, only another GAA-TTC site can bind. No other enzyme site can bind. Only GAA-TTC can bind. কেউ bind করতে পারে শুধু GAA-TTC. Only this. It can be from another DNA. So if you cut a vector with EcoR1 site, it will also have a AATTC overhang. So this interesting from this can be inserted into a vector. So this is actually, let us look at a vector below. A vector has always got a site, and that is known as restriction site or multiple cloning site, you can call them. MCS contains restriction enzyme. Restriction enzyme site. It will contain also EcoR1 site, depending on the vector. It can have a He3, BAM-H1, whatever. It will have a restriction enzyme site over here. So this particular restriction enzyme site you have, and this vector also needs to have a sequence known as origin of replication. This is the origin of replication. A promoter region is there in many places. Just in case you want to transcribe and translate this DNA, this is an insulin gene, say for example, you want to transcribe and translate, make that protein. So you need to, it has to be under the control of a promoter, which is ahead, upstream. So you have keep a promoter sequence over there. And very importantly, you must have another gene. This is a antibiotic gene. This is an antibiotic gene which is present over there. This can be ampicillin or whatever. So, when you transform this particular vehicle or vector in a host cell or bacteria, E. coli, that bacteria must be resistant to this particular antibiotic. So, you can distinguish between cells which have been transformed and which have not been transformed. You grow them in a medium which has got ampicillin, this antibiotic. Ampicillin, tetracycline, you can use any antibiotic you want. You just grow them over there. The reason for this being that you need cells which contain this plasmid. The cells which don't have this plasmid does not have resistance to antibiotics. So this is a very important thing which a plasmid must contain. And now you cut this also with a restriction enzyme, say GAA-TTC, one example, and this is your gene of interest or DNA of interest. You can call this as DNA of interest. This is the restriction site. Restriction enzyme site. जुड़े गए. Overhang, you cut it with the same EcoR1. This is also EcoR1 cut. N. Okay. Now you have put it inside this. Since it's EcoR1, they will match and they will join. So you can cut this and paste it inside. And you have the promoter region. For replication inside the cell using the host machinery, you must have the ORI sequence. This is the origin of replication sequence. If this doesn't happen, the plasmid won't be able to replicate. This sequence is needed for plasmid replication. And of course, you will have your antibiotic resistance gene. This is antibiotic resistance gene. So you have created a vehicle, a vector or a vehicle, vector or vehicle. In parasitology, vector means something, it carries the disease to another organism. From mosquito acts as a vector. It takes this and transfers the dengue inside you. So mosquito becomes a vector. Here it is a DNA sequence in molecular biology. So you have a vehicle which contains a gene of interest, say for example insulin. It has got a promoter. Because of this promoter, this is going to be transcribed and translated using the bacteria or the host machinery of transcription and translation. So the plasmid inside the bacteria is going to get expressed because of this promoter region. And because there is this origin of replication site, the bacteria's replication machinery will go and bind and cause this plasmid to replicate inside the bacteria. So using the bacteria's endogenous replication, transcription, translation machinery and making this particular gene of interest or insulin, and it is replicating, and you are screening by growing the bacteria in antibiotic resistance media, and because of this gene, this particular thing is selected. So those who don't have this particular vehicle, they will die. They will not grow in amphiphilin. Only the ones which have got this antibiotic resistance gene, they will survive. So what you do, you transform them into bacteria. Transform into bacteria. Some call it the host. Host cell. And now the funny thing is, after screening for antibiotic resistance, all cells which have not been transformed by this plasmid, they will die. Only the cells which have got in the antibiotic media, which have got this antibiotic resistance, they will survive. Clear? So, and it is going to use the bacteria's machinery and make more and more copies of, say, your gene of interest or insulin. Now your work is, you break open the bacteria and isolate and purify the protein of interest, insulin, and you bottle it and sell it. So human insulin, those who have diabetes, they can take insulin injection in their stomach and they will stay fit. So you don't have to slaughter thousands of oxes. And former times, before this recombinant DNA technology, my grandfather, they used to take ox pancreas insulin. So all these in the slaughterhouse, the oxes which are oxes or goats or cows which have been slaughtered, they will take out their pancreas, and that from that they will make extract and take out insulin. It becomes very expensive. Now these bacteria, overnight you can grow liters and liters, as much as you want. Just one of this transformed cell inside, grow them, and the next morning break open the cells, purify the insulin, bottle and sell it. It becomes very cheap. My grandfather, when he used to take insulin, one insulin, something for 1 month he used to take. This much used to cost 40 rupees, and this was in sometime in 1980s. Nowadays it can be near to 10,000 or something. If you isolate it like that, and strict BJP rules, even this will be banned. We are killing cows for insulin, and all these will happen. And so all these problems will start taking place. But now, even after all greed and profits and everything, this insulin costs 500 rupees a vial for a month. Now today, after all greed, actually the production cost is less than 50 rupees. Government tax, iska cut, ye gunda ka tax, ye gunda ka cut. The doctors going medical going for a Switzerland vacation, सब कुछ लगा के. After putting all that, it's coming to 500 rupees. ये 50 rupees 10 times price में 500 rupees में बिक रहे हैं. And guys go to more expensive companies, they will charge this one for 1000 rupees. उससे ज़्यादा ले ही नहीं पा रहे. They are unable to take charge you more. क्योंकि लगता ही नहीं है. जो चीज 5 रुपया में लगता है, now they are taking at 500 rupees. 50 rupees or 500 rupees. They cannot even take more. They are trying different methods by which they can charge more. This VAT, this tax, this GST, this all these. They are trying to do it because the greed of a government is the reason for recession. Recession या job market down, it doesn't happen normally. जब हम लोग का अपना सरकार दे बिकम ग्रीडी, जो चीज 5 रुपया में बिकता है उसको तो 10 रुपया में बेचो, उसको वो लोग 50 रुपया में बेच रहे हैं. एंड वेयर इज द मनी कमिंग? इट इज कमिंग फ्रॉम योर टैक्स पेयर्स मनी. दिस बिग इंडस्ट्रियलिस्ट, दे आर टेकिंग लोन्स फ्रॉम बैंक. नॉट ओनली लोन्स, ये अंबानी, अदानी एंड ऑल दीज, द रिचेस्ट मैन. He takes crores and crores of loans. He takes loan from the bank to buy the bank. बैंक से लोन लेता है बैंक को खरीदने के लिए. बैंक को खरीद लिया, and what do they do after that? You know what happens? Bank then कुछ दिन बाद government will write up the loan. जो लोनेर मुकुर हो जाए सब. That from where is the money coming? This is the public's money. Anyway. So, science is bringing out things, methods by which you can make it cheaper. But nothing can get above human greed. Okay, so let's go to the next part. I myself made this. I know how cheap it is. If you use it very properly without wasting and all these things, you can even make it as low as 5 rupees instead of 50 rupees. I'm talking about using the best quality, paying all the scientists the required money and everything. After taking that, it's going to cost you 5 rupees without wasting anything. Take profit of everything. You can sell it for 50 rupees. But why you are selling it for 500 rupees, I don't understand. If you see, take one of these transformed cells, suppose this is a bacteria, this is the host chromosome inside, bacterial chromosome, and you have these plasmids, they are replicated inside. They are keeping on producing your gene of interest. Okay, yes, tons and tons. They are also so this is you're growing it in a flask. It has got a medium known as LB medium, Luria Bertani medium. Sometimes it's called a lysogenic growth. They are growing it in a special media for bacteria. It's got peptone, tryptone, yeast extract. All these things are there. It's very rich for the bacteria. Every 20 minutes, one cell becomes 2 cells. In E. coli. So, today at 6 o'clock if you put one cell, So 20 minutes, one cell will become two cells. Next 40 minutes, two will become four. Four will become eight. Next day morning, you have billions of cells over there. And then when you take this cells, you lyse them and purify the extract. Purify the protein extract. There are methods. Some ammonium chloride and all these methods. You will have tons and tons of insulin. 1 milligram insulin आराम से आ जाएगा just from 50 ml flask of E. coli grown. 1 milligram of insulin, that is something over 1000 units, and one vial which costs they are charging 1500 rupees, it has got 300 units. So 3 vials में 1000 units बन जाता है. So in 3 vials you get 1000 units. So एक flask 50 ml flask hardly media price will be hardly 2 rupees 3 rupees, and isolation 5 rupees. You will get 3 vials. So it is very cheap, but there is nothing for taxes and all. वो अपना until unless we clean ourselves inside, there is nothing can be done. When I have to pay so much money, I feel that I have made these things. इतना सस्ता होता है. Why are they charging so much? Okay, so there are 4 major types of vectors. First is the plasmid, which we will study in detail. Plasmid. Then you have viral vectors. Then you have cosmids. And then you have artificial chromosomes. What are these artificial chromosomes? Bacterial artificial chromosomes, yeast artificial chromosomes, mammalian artificial chromosomes. Chromosomes, they are making chromosomes where you can gigabytes and gigabytes of data you can put inside. In computer terms, so many gigabases of nucleotides you can put, so much sequences you can put in. This is like a chromosome. You have yeast artificial chromosome, YAC, mammalian artificial chromosome, MAC. You can grow them in mammals, you can grow them in yeast. Yeast, YACs are very popular. So common to all the vectors, what do you have? You have an origin of replication, which is where it has to be there. Origin of replication. It will have a multiple cloning site, MCS. Multiple cloning site is the site where there are lots of common restriction enzymes are there. So common, the EcoR1, Wnt3, BAM-H1, all these enzyme sites are there. So depending upon your insert, you can use that particular any of these restriction sites and you can clone them. Cloning, Zaka madam will take it later in details, but depending on this, you can use it. Then it should also have a selection marker. Like antibiotic resistance gene. It should have. In yeast you have orthotropic mutations. So you grow them in a media where this yeast cannot synthesize tryptophan. So you have introduced a plasmid where tryptophan synthesis can be done. So in a medium where there is no tryptophan, the ones which don't have a selected transformed, the non-transformed cells, they will die. Only the ones which have been transformed with tryptophan producing genes, they will survive. These are all nutritional mutants they are doing. So now let us come to plasmids. So what are plasmids? They are double-stranded. We are talking about plasmid, double-stranded, generally circular, not always. They are generally circular DNA sequence capable of automatically replicated autonomously, capable of autonomous replication in host cell. Okay? So the plasmid has been transformed in the host cell, but it must be capable of making more copy numbers of itself. দুটো দিলে তো দুটো generation পরে দুটো তো will go away. So you need to have, it needs to keep on replicating. It has some capabilities. Autonomous replication, one of them. They must have compatible and common restriction enzyme sites. If there are no compatible and common restriction enzyme, বাজারে যেগুলো পাওয়া যাচ্ছে normally available restriction enzyme, if the sites are not there, then how will you put the insert inside? You need to do it. You need to find. Then there are a lot of drawbacks which we will talk next day. They must be small in size and able to carry large inserts. Able to carry large inserts and selectable markers. So the size range is normally 1 kilobases to something like 250 kb. Normally, an ideal plasmid which we normally use is around 3 to 3.5, 2.5 to 3 kb. The bigger the plasmid becomes, the more difficult it is to transform it into cells. It's easy, the smaller it is, better. And 1 kb যদি plasmid, if the size is 1 kb and the insert is 2 kb, size of insert is greater than size of vector. So it becomes very difficult to put it inside the transform it. So something like 200 to 300 bases are easily cloned inside it. But therefore the normal size is 3 kb around plasmid range. They encode only few proteins required for the replication, and they are located near the Ori sequence. The rest of it for replication they take it from the host. Host का machinery ले लेता है, bacteria का machinery ले लेता है. Plasmid में there are some sequences when it's transformed inside it, it starts to produce its own protein. for replicators, only some of them, but most of it they take from the host. So basically if you draw, this is a bacteria, this is a bacterial chromosome, or it's not exactly like a chromosome. This is the bacterial chromosome, you can call them like. And here small double circular plasmids. These are the plasmids present inside. So the host range, which is host range is determined by the Ori sequence. Determined by origin of replication. If it is of the col E1, it is the bacterial origin of replication, col E1 from E. coli. They will be able to only replicate in bacteria, enteric bacteria like Salmonella, E. coli. They will not be able to grow in other places. But so depending upon which particular, if it's of RP4 type, they will grow RP4. They will grow in all gram-negative bacteria. ঠিক আছে? I am wiping this. Now, these particular plasmids, why will a bacteria take a plasmid inside itself? Why will it need a plasmid? It gives some sort of power to the bacteria, extra power. Okay, like in Brahmastra, you wear all those things and you get power. It gives some extra power. I was seeing Brahmastra this Monday. I saw it. খারাপ লাগেনি, ভালো লেগেছে বলতে হবে। Apart from the stupid romance and love, the rest of the movie concept was good. Okay. So what are the powers which gives? First thing is, it gives antibiotic resistance. It's a big problem nowadays in hospitals. In hospitals, it's a big problem nowadays. These bacteria, they are resistant to all sorts of antibiotics. Why? Because they have developed these plasmids. যতই antibiotic দিয়ে যাচ্ছে, people are still getting infected and dying. These they become super bugs. So first thing is antibiotic resistance. Next is antibiotic production. It will produce some antibiotics which will kill others. There are some aromatic compounds like phenolic compounds in the surrounding. So these plasmids will bring in enzymes which will degrade these aromatic compounds. These aromatic compounds are not good for growth of the bacteria. In the environment, if aromatic compounds are there, so they will give the power to degrade aromatic compounds. Degradation of aromatic compounds. Then hemolysis. If it's a blood bacteria, it will cause the blood to erupt. Hemolysis. It will break the blood cells. Sugar fermentation. Very important. मौद बना है, making alcohol, sugar ferment करे. Enterotoxin production. Because of these enterotoxins, you hold your stomach and run to the toilet. These enterotoxins, they will cause irritation in your stomach. पेट खराब. Heavy metal resistance. हमारा नेक्स्ट क्लास शनिवार ना, नेक्स्ट क्लास बैक्टीरियोसिन प्रोडक्शन. Other bacteria that used to kill. It will kill other bacteria. Very important, which we use in plant transformation, induction of plant tumors, agrobacterium. Then hydrogen sulfide production, H2S production. You have them in spring bacteria, hot thermal spring bacteria, sulfur producing bacteria. Okay, next year we will continue with the rest part of plasmids, and then from plasmids we go to cosmids. ওটা হয়ে গেলেই আমার পাঠ শেষ.